So without further ado, I'm going to go ahead and introduce you to Corinne. Corinne, do you want to introduce yourself and tell us what your title is?
Sure. Hi, everyone. My name is Corinne Fuchs. I'm the Marine Invertebrate Collection Manager at the FWRI Florida Biodiversity Collection.
All right. And before we get started, I want to introduce you to Paul. Paul, go ahead and introduce yourself.
I'm Paul Larson. I am the curator of the collections at the Florida Biodiversity Collection at FWRI.
All right. So you guys have a presentation for us today?
Yes, we do.
All right. Go ahead and cue it up, and then I'll send it live.
All right, looking good.
There we go. Everybody see that? Corinne, when you're ready.
Great. Okay. Hi, everyone. Again, thank you for joining us. So hopefully a lot of you have been to museums where you can walk around and look at exhibits and learn about, weird animals and everything else in the collection at that museum. But behind the scenes of those museums usually is a collection with thousands or millions of items that aren't on display out on the public floor. Unfortunately, our museum doesn't have the space for public exhibits, but our collections are big and are used by scientists and other researchers all around the globe. So researchers deposit specimens with us as vouchers, physical proof of the species that they were studying. We at the collection preserve these specimens so that scientists today and in the future can study the animal to learn about where and how it lived. So today we're going to be looking at some digital models of actual specimens from our collection that our partners at USF Tampa have scanned that came from Florida waters just off our coasts. So we'll start with the familiar, which is the chordates. Chordates are a group or phylum of animals that have backbones. So you and I are chordates, fish are chordates, birds are chordates, anything with a backbone. All chordates have three important features, although they can actually look pretty different in different species. One is a nerve cord, so that's just like our spinal cord, our nerve cord running down our back. One is a tail, which of course in a dog you can see very clearly. In a human, it's just a bunch of tail bones at the end of your spine. Another is gill slits, which we clearly don't have gills, but those slits will become part of our jaw and throat as we grow. And finally, a notochord. So that's a rod that supports the body. It's what becomes our backbone. So chordates are really big and common on land, so you've probably all seen a lot of them. But there's a lot on the planet, and there's a lot in the water too. So we're going to start out with this dusky shark jaw, which is from, again, off the coast here. But what makes this a specimen and so special is if you zoom into that corner there with the white tag, you see there's a bunch of writing on it. So on there we've got a few things. We've got Carinus obscurus, which is the species, the length of the shark that this jaw came from, the width of that shark, the gender, the male, the date it was collected, and then we've also got some codes with numbers and letters that we can put into our database to look up even more info, like who identified that this shark was a dusky shark, and when exactly was it collected, and when was it put in the collection, and all sorts of stuff we can't just fit on the tag on the specimen itself. So without this data, it would be a lot less useful for science. If you zoom out, you can see this kind of looks like bone, and shark, like all fish, are chordates, but sharks, rays, and skates and their relatives are special because unlike us, their skeleton is made of cartilage. So cartilage is the soft, flexible stuff that keeps your ear structured, but still bendable, and your earlobes. But if it's thicker and stiffer, as it is in the shark jaw, it's actually really structural, but still pretty flexible. So here, everything you can see in this jaw is all cartilage, except for the teeth, which there are a lot of. If you zoom into the teeth on the lower jaw, you can see that the teeth are very broad at the base and then very pointed at the tips. And then if you zoom into the teeth on the upper jaw, those teeth are a lot more triangular. There's mostly one angle going on there, just kind of going to a point. And that is so that this shark, a dusky shark, can eat animals that are too large for it to just swallow. They can kind of move back and forth to tear bits off of their prey to make them more mouth-sized. But this isn't the only types of teeth sharks can have. There's a whole bunch, including like piercing needle-like teeth used for really fast-swimming fish to keep them in the mouth, and stuff like crushing teeth that will smash into clamshells and other hard prey. And finally, if we look at the back of the jaw, the teeth sticking up that we saw from the front, there are a lot more back here. Usually, if the shark's alive, you can only see that first row that's sticking up that we saw from the front, because it'll have gums and tissues like we do, covering most of those rows of teeth. But each row of teeth there, all those series of teeth, continuously grow. And as that first row loses or breaks teeth, a new tooth will come forward, rotate in, and replace it, so that the shark always has a good set of teeth. So that's our shark. We're going to go to a somewhat more normal fish. I say this because these are from a group of fish that have bone skeletons, hard skeletons, like we do. But this is also a pretty weird fish in itself. So this is a Peacock Flounder. It's a flat fish that lives along the seafloor as an adult. It starts its life out looking pretty much like a normal fish. It's got an eye on each side and a fin on each side. It's a fish. But as it starts to grow, one of its eyes will actually move around its head until one side of the body has both eyes on it. We call that side the eyed side. Here, the number 2 labels the eye that migrated from the other side of the fish, where the number 3 indicates the one that was always on that side. It's kind of like in the normal place you'd expect a fish's eye to be. The eyed side here, right, has both the eyes, so the side that doesn't have any eyes is the blind side. And that's the side that's facing the ground as the flatfish swims sideways against the sea bottom. So eyed side will be looking up, blind side will be facing down. The eyes aren't the only difference between these two sides, though. Label 16 marks the pectoral fin, and we're looking at the blind side, so it's that little 16. But if we rotate around to the front and look at label 7, that's also a pectoral fin, this time on the eyed side. So it's super long and pretty stiff, and actually really handy and useful on this side for the fish to swim and maneuver, while the pectoral fin on the other side is shorter, because if it were this long, it would just hit the seafloor anytime the fish tried to swim. And then finally, we can't observe the notochord or spinal cord of this specimen since we're outside it, but we can see some other stuff that is unique to chordates. Gill slits in fish actually develop into gills, so we can see the dark slit is the edge of a protective structure marked by 4 and 5 that protects the gills. So mouth will open, water goes in the mouth, water goes over the gills, water comes out of that dark slit. And then we can also see, if we zoom back out, the fish's tail. So in this case, it's a caudal fin, but it's a finned tail. So that's its postanal tail. All right, moving along to invertebrates now. We're going to start with mollusca. So mollusks have 3 major traits as well, very different from chordates. First is a mantle, which is something that usually makes a fluid that layers and hardens into a shell. So clamshells, all that stuff you see, this big shell here. As the animal grows larger, the mantle grows larger, and so does the shell. So a lot of mollusks are slower, soft-bodied animals. They really rely on these shells for protection, but there are some groups like squid and octopuses that have actually lost their shell and instead use speed and their ability to hide and mimic and camouflage to protect themselves from predators. All mollusks also have a muscular foot that they use for locomotion. It can be a lot of shapes, such as just your typical land snail that you might see in your garden, just a foot. It can also be a tongue-like foot that clams will use to bury themselves in the sand, or even for octopuses, split into multiple feet, like the 8 arms of an octopus, so that they can handle objects and lined with suckers to make it easier to manipulate stuff. And then finally, most mollusks also have a radula, which is a hard, rough structure in their mouth that they use to feed. So the radula might be specialized to scrape algae from rocks, or it can even be super, super specialized into a harpoon-like structure to sting prey. In this lightning whelk, this specimen is just a dried shell, which makes it easier for us to store and cheaper for us to take care of than if we had cut the entire animal. However, there is a trade-off, because if we keep the whole animal, then we can get a lot more information out of the animal in the future. So we try to keep both wet and dry specimens as we can in the collection. In most snails, the hole or aperture that the snail can retreat into and hide back into its shell that aperture is usually on the right side in snails. In this one, it's on the left. So if you put your left hand up to the screen and try to grab inside the shell, your left hand goes in and not your right. This is called sinistral, or left-handed. If we rotate the shell to view the apex of the tip of the shell, we can see coiling going counterclockwise from smaller inner whorls to larger outer whorls. And if we rotate back to that aperture view, inside the aperture, there's a dark brown oval marked with a 3. This is a hard, flat plate called an operculum. So in life, it's attached to the snail's body and is used as a door to protect and seal the snail when it retracts into its shell. And in some species, it's actually even used as a weapon. And then if we zoom out, this model looks pretty small on a computer, right? But lightning whelks can get very large. Very large. They use their heavy shells as weapons. They'll actually pry open or grind down their prey's shell using their big heavy shell. And once it's exposed, they can get their radula inside and scrape out the prey and eat their meal. All right, then we've got arthropods. Arthropoda is the most numerous and diverse animal group. There are about 1 million described species we know of, including shrimps and insects and scorpions. All arthropods have an exoskeleton, which is a hard outer covering. It doesn't grow with the animal like a mollusk's shell does, though. Instead, when the animal gets too big, it has to molt or shed this skeleton. That reveals a newer, flexible skeleton underneath that the arthropod then can fill with water to make it grow larger, and then waits for it to harden. Then it's got some room to grow until it has to molt again. All arthropods also have jointed legs and other jointed parts. Arthropoda actually is jointed leg because with that hard exoskeleton, they can't flex things like we can. With those joints in place, though, they have enough range of movement that they can easily move eye stalks and antennae and even the body itself. And arthropods can be found in almost any habitat, from the deep ocean to the tops of mountains. If you've ever overturned a rock and watched little armadillo-like critters or pillbugs scramble for cover, those are isopods. Isopods can also be found in the deep ocean, and some species get really big, like this one. So this one on screen is a giant deep-sea isopod. It's about the size of a football, but it's still flexible enough to ball up like those little isopods on land can do. The dark lines along its body mark the joints in between segments that allow it to fold. And these are all scavengers that feed on dead mammal carcasses. So whales and dolphins that sink to the bottom of the sea floor, they find them. If you rotate the specimen to look at the head, you can see the antennae, marked number 3, which they use to smell out those carcasses and find. And then it's also got mandibles on either side of marker number 5, those little hooky things, that allow it to bite off pieces of food from those big carcasses. And then finally, moving to the rear, label 9 shows 7 jointed legs, and behind those legs are some darker brown flat stacked parts we call pleopods, number 10. These are for swimming, so when the isopod flutters these back and forth, they actually paddle the isopod above the seafloor despite its heavy body. And lastly, we're going to talk real quickly about echinodermata. Echinoderm means spiny skin, so the animals in this group have well-developed spines covered by skin. This is sea urchins, sea stars, brittle stars, and sea cucumbers and the like. Most phyla, most animals, exhibit bilateral symmetry like us, where we have a left side and a right side mirrored. But aside from a few exceptions, in echinoderms, most echinoderms are radially symmetrical, so they're symmetrical circularly kind of around a single point. They also have a system of water-filled canals that hydraulically support them and push water into and out of their tube feet to move their feet. These usually have suckers on them as well, so the animal moves by coordinating a bunch of, like, hundreds of little suckered feet on the bottom of the sea. This one we're looking at is Oriaster reticulatus, and we can see those tube feet extending from this groove on the middle of this arm, on the bottom of the animal. This animal has pentaradial symmetry, which means penta, 5, so there are 5 of these grooves actually extending out from that middle of the disc there, which is where the mouth is. So that mouth is facing downward as well to feed. Some stars can swallow their prey whole, but this species will actually evert, so put out its entire stomach on top of its prey, wait for it to digest outside its body, and then pull the stomach and the prey inside it all back up, already digested. Juvenile cushion stars, so the species are eaten by multiple fish species, but as an adult they can get really big, 20 inches or so across, so few things are big enough to eat it. But adults are also usually really bright, like a bright orange, bright red color, so they're easy to see from boats, especially since they live in shallow waters, and people have overharvested them, for decorations across South Florida and the Caribbean. So knowing this and seeing them struggle, they're protected in Florida waters. It's illegal to collect this species, even if you have a fishing license, so that the species has a chance to recover. And that was just, a handful of the specimens we have in the collection. And we're even just looking at models, not the specimens themselves. So we can learn a lot about the live animals that these represent and how they lived and survived in crazy conditions, and look back in time when we get better technology to learn even more about these animals and how they lived in the past and now.
Thank you so much, Corinne. If I can get my helper to share the screen so we can get all of us up on there, then we can answer some questions. All right, thank you again. It was really interesting. I love the 3D versions of these specimens. All right, a couple of questions that have come in. Do you have sharks in your collection? You had shark jaws. Do you have actual sharks?
Paul?
We do have actual sharks in the collection. We have 3 collections actually in our museum, and one of them is all for fish and fishy-like things, and it does include some sharks that are intact with all the tissues and everything on them. They just look like a preserved shark, and they are stored in some large metal vats because they're too big for a jar.
And how many specimens do you guys have?
In the invertebrate collection, we have 139,000 lots of specimens, and each lot is a single unit that could have multiple specimens within it. So right. The truth is we don't have the data for all of those, so I can't give an exact number, but we have a lot of specimens. Yes,
you do. So why is it important to keep a collection like this?
So there's many reasons, but one is just to understand biodiversity. Biodiversity is the sort of spread of all life, the diversity of different species and different animals within those species on our planet. And greater biodiversity actually keeps ecosystems doing tasks for us that are helpful, like filtering water and preventing storm surge during hurricanes. So the more biodiversity we have and the more we can track that, the better we're able to predict how things will respond to big things like storms. In addition, we also, having that huge collection now means that if we get better technology in the future, we still have stuff from the past that we can use that technology on. So if you think about genetics and DNA, we really didn't understand it until a few decades back, whereas now we can go back and do a bunch of genetics work on specimens from before we knew how to do genetics work.
I'd also like to add that having a collection like this allows us to discover new species. We've had collectors come out and didn't realize that they had things that were new to science, and they deposited in our collection. And as we examined them, we realized that these were brand new species to science, undescribed, and we give them a name and they become officially part of our known world.
That's amazing. So do you guys have specimens just from Florida, or predominantly from Florida, or do you have them from all over the world?
Predominantly from Florida, but we do have a smattering kind of worldwide. I would say at least 90% of the specimens are from Florida or the waters around Florida, nearby, Gulf of Mexico.
All right, we have a question. What is your favorite specimen in the collection?
My personal favorite is the blanket octopus. We have a few blanket octopuses actually, but these are very large purple octopuses that they swim offshore out in the open ocean, but they don't live very long, and they occasionally wash up on shore, and that's basically the only way we can get them. So those are kind of neat and unique.
And I laugh because this changes every time someone asks me. There's just so much cool stuff, but I think today it's we've got a bunch of really cool sea spiders, both from Florida and from Antarctica, where they get really big. And these are these crazy little animals that have four pairs of legs like a spider, but they're so small and they have so much leg that they don't have any sort of lungs. They just kind of exchange gas over their skin.
Wow, that's pretty amazing. All right, so for our students and teachers and everyone on this call, I'm going to ask you both the same question, but you know, what kind of, how did you get into this career? You know, were you always into the science? Were you into something special? And then like, what kind of background or education do you need to get into this career?
I'll go first. So I've been interested in science for a long time. In elementary school, I started to get really interested in it. And then around middle school, I think I started really focusing on biology as what really kind of held my attention. For the job I have, I went to college and I got a bachelor's degree. And then I went to graduate school and I got a PhD studying marine invertebrates in particular. And then after I graduated from there, I was qualified for this job, and when the opportunity came up, I applied for it and was lucky enough to get it.
So similar story actually. I grew up in Florida, running around the forests and in the water, so I really loved sea critters, especially for my young age. I didn't realize that science was kind of a job that I could do for a long time until I took a field course my first year of college and realized, oh, not all science is just like a bunch of syringes and test tubes. There is that, but there's also, weird fish and weird critters and stuff. So I knew I was interested in biology for a long time, but once I took that course, I went, this is it, I want to do this. So I got involved in research in undergraduate, got my degree. And then I also started graduate school, which I actually haven't finished, but I'm a good chunk through. And that qualifies me for this job that I thankfully got to apply for last year. And it's been really wonderful so far.
Well, thank you both so much. I think we're going to wrap up our questions here.